Very good morning everyone. Myself Suraj Vaide from Nepal. First of all, I apologize to all that I could not attend the program physically due to the prolonged visa process. Till today I haven't received the visa. With your permission, I would like to share my slide in audiovisual form. The ecology of the wheat cultivation in Nepal. There are two seasons of the wheat cultivation. Main season is planted on the October and harvest in June, and another planting season at the high hill areas as summer wheat crop on June and harvest on the October and November. It means year-round wheat is cultivated in the country. Here you can see that more than 50% of wheat cultivated areas fall in the plain areas, and it contributes nearly 2/3 of the total national wheat production. Likewise, 35% of total wheat at hill and contributes around 30% of total wheat production. There are major biotic factors which directly impact on the wheat production, and their incidents are also vary with different time interval. Here you can see that all tree rows are increasing trend since last decades. Similarly, the foliar blight and powdery mildew are also increasing trend, whereas the bond and the loose spot are decreasing as compared to last few decades. When we talk on the importance of CIMIT programs on crop varietal development and management of the disease in the country, it plays a significant role on the disease monitoring and pathotypes identification, disease early warning system, germplasm exchange, their screening, capacity building training. Detection of the yellow loss virulence pathotypes, the PST6, PST16, UG19 and variants, TTKTT are an example of the monitoring of disease in the country, and also the valuable support from the Abboud Ross Analysis Laboratory. What we did on the wheat disease monitoring in the last year in Nepal. In total of 216 different wheat fields were monitoring at 45 districts from the 7 provinces. It was done from the lowest altitude of 59 meters to the 2,844 meters from the sea level. It almost covered the major wheat growing areas of the country. The yellow rust disease usually incident at the high hill areas and around 18% at the plain areas. It was not recorded at the plain areas last couple of years before. It might be due to the incident of the heat tolerant pathotypes prevalence in the country. Around 2/3 of the sample recorded at a moderate to high level of disease severities in case of yellow rust disease. Similarly, in case of the leaf rust disease in the country, it appears mainly on the plain areas, whereas 23% at the hill area. A decade before, leaf rust was not recorded at the high hill. Now it observed in the hill. It might be due to the increasing temperature and genetic diversity. Leaf rust is also increasing at the high areas also. More than 70% samples shows the moderate to high level of disease severity. Steam rust mainly appear at the maturity stage of the crop and low at the moderate level of the severity. It is commonly recorded in the high hill areas at May to June. At this time, most of the wheat crops has been harvested at plain areas. Here you can see the field where UG19 variant was recorded in the last November 2023 at Dangdunge and Kharidunga of Dolkha district. The variant TTKTT was confirmed by GRRC Denmark. The sample were collected from the summer wheat crops where the farmers are grown the wheat as a green fodder. The crop were also cut off after the 15 days of samples collection for the animal feed. Then they are planted a potato and other vegetable in the field instead of wheat crops again. Then after we were regularly monitoring the wheat field across the country in main wheat crop season, the UG99 was not detected again from the wheat field. Even in the last 2024, there was not recorded the steam rust at that place as well. However, the leaf rust and yellow rust were detected in that areas. It In main season crops, other race belongs to Clade IV-F groups, that is TKKTF and TKFTF, were detected in the samples from the western, midwestern and center part of the country, analyzed from the GRRC lab and also from the marble technique in our lab at Kumoltar. The same lot of samples were also sent to the Indian Institute of Wheat and Barley Research, ICAR, Simla, India. They were pathotyping them as 75G5. On the basis of disease monitoring data information, we also provided advisory services for the early warning disease system with collaboration with the Met Office and University of Cambridge, UK. In the advisory services, we mainly advise to the farmer on the early warning disease forecasting, appropriate date of planting, selection of genotypes with respect to their location, time of field observation, awareness on the potential outbreak of transboundary diseases, application of fungicide at a hotspot area where the early detection of the disease. These information are provided to the stakeholder through the different mode of dissemination, such as mobile SMS, WhatsApp networking groups, agromet advisory bulletin. This year we have targeted to disseminate the information to the 0.2 million client. Here the table shows how the information flow to the client and who are the responsible organizations and key contact role on the dissemination of the information. This is the flow charts of the WhatsApp groups, how the message goes to the client from the admin. There are 3 steps of stakeholder who are receiving the information and deliver the corresponding to their client. First, the admin disseminate the agriculture extension personnel of the municipality and agriculture knowledge centers. Then they send the message to their leader farmer who are connecting their respective municipalities. In the next step, the leader farmer are disseminating the message what they receive from the agriculture extension personnel, and the feedbacks from the client are also received by the admin through the same pathway. There are the messages to send the client through the different mode of dissemination in local Nepali language. The promising and pipeline genotypes also tested the international genotype screening platform at Carlo, Kenya, against the different Ross pathotypes. In the last year, 160 Nepalese wheat genotypes were tested in Kenya, including 21 most popular released genotypes in order to expose them to different variants of UG99 and other races of yellow and steam rust pathogens. From the screening data, it seems most of our released and popular genotypes are vulnerable to yellow and steam rust pathogens. Very few of them shows the resistant to moderately resistant to the pathogens. So it shows that there are the prompt need to release the diversified varieties in order to protect the crops from the potential transboundary pathogens. Similarly, in case of the pipeline and promising lines, less than 10 and 20% lines are resistant to moderately resistant to yellow and the steam rust pathogen respectively. It means there are also need to introduce the exotic line having the different genetic background for further breeding programs. We also do the regular screening our genetic materials in natural and inoculated condition at different agroecological location of the country. Out of thousands of lines, few of them still performing resistant to different pathotypes of the leaf and yellow rust pathogen. We usually do the inoculate the pathogen from the mixture of pathogen collected from the different part of the country, because the selected resistant lines from these tested line will be released for the different agroecological zone of the country. So they need to expose them in high population pressure of the mixture pathotypes during screening. Here you can see some effective and ineffective YR genes against the different pathotypes of the yellow rust pathogen in inoculated condition in Nepal. It's recorded from the wheat yellow rust differential as well as international wheat disease trap nursery for the yellow rust disease. In last 10 years, we supported from CIMMYT Mexico, we could success to release the 13 wheat genotypes, including with 6 biofortified wheat genotypes. Here you can see in the table. For chemical option, we are also conducted the efficacy testing of different fungicide. Here you can see in the table that application of propiconazole called TILT at the rate of 1 ml per liter or tubuconazole plus trifloxystrobium's content product called Nativo at the rate of 0.5 gram per liter of water in the 3 times at the 14 days interval were more effective to control the disease than the other chemicals. Now we have also facility to conduct a multiple diagnostic techniques for the yellow and steam rust pathotype in Kumultar. We sincerely acknowledge to Dine and from the zone in Center UK for the continued supporting to run the techniques in Kumultar. Regarding the centennial plot experiment, we established the experiment in both summer and main season wheat crops at 5 different locations. 2 experiments for summer crops and 3 for the main season crops. The experiment have been represent all the altitude from the plain to high area. In the summer season, it was conducted in the Mustang and Dodeldura. In the main season, it was conducted in Kumultar, Dolkha and Bhairava. Now, in the experiment, it shows that no steam rust disease was detected in all locations. In Dolkha, 6 lines shows a susceptible reaction on the yellow rust disease, whereas the 4 lines shows the susceptible to leaf rust disease. In Kumultar, 13 out of 24 lines had susceptible to yellow rust disease. 4 lines had the leaf rust susceptible. No yellow rust was recorded in Bhairava. In Dolkha, out of 24 entries, 14 lines had infected by powdery mildew disease. Since last couple of years, powdery mildew disease is increasing trend in the high hill areas and hill areas as well. Dolkha was one of the hotspot of the yellow rust disease, but it was now become a hotspot for the powdery mildew diseases as well. Here you can see the picture reflect the disease monitoring of barberry bushes, grasses and wheat plants at Nagarkot, Goragori, Zidi and Dolkha have been carried out in presence of Dr. Daves, Dr. Mary Celis and Yusuf in different time intervals. Here you can see at the center of the picture showing the monitoring of barberry bushes by Mary Celis from the sitting in the car at Goragori. There are the series of barberry bushes along the site which can be observed from the vehicle as well. The collected barberry sample were inoculated in the National Plant Pathology Research Center, Kumultar, with following the Dr. Eugene protocol. There were no infection of the wheat, even spores were landing on the leaves. The experiment is continuing the coming days as well. DIWAS is a program is supporting on the early warning alert information about the early control disease mechanism in the country, which will help to stop the inoculum spread in the new areas. Technician and wheat growing have become aware on the timely control the mechanism with application of systematic fungicide in the disease prone area. Support on the continuous monitoring, message alert through the SMS, advisory bulletins and other e-news to concern the stakeholder, including wheat grower, extension technicians, NARC scientists for the early warning advisory system network. Also support on the successfully developed and released a considerable number of the wheat genotypes having desired traits such as resistance, biofortify, high yielding, etc. A need to continue networking system on the early warnings advisory information to the different stakeholder, regular monitoring and action plan for mitigate the rust pathogen in the hotspot area timely, introducing the advanced genetic materials for the deploying the durable wheat resistant genotypes, capacity building training and exposure to scientists on the advanced plant breeding techniques, assessment of different wheat diseases, regular evaluation of Nepalese germplasm, international phenotyping platform against the potential transforming disease such as rust and blast. And acknowledgement to the different organizations for their valuable support in terms of funding, technical backup, capacity building, training. For further information, you may contact me through the mail. Now, Dr. Dave will also assist if you have any queries. From here, I request to the Dr. Dave. Thank you.